The overall goal of the following experiment is to assay thermal nociception in Drosophila larvae. This is achieved by harvesting Drosophila larvae to subsequently expose them to noxious thermal stimuli. As an optional second step, larvae can be UV irradiated, which causes tissue damage and thermal nociceptive sensitization. Next, larvae can be probed locally with a heat probe or globally using a heat plate in order to provoke nociceptive responses that can be quantitatively measured. The heat probe shows that larvae have a ceiling to their nociceptive response to heat and a surprising relationship between input temperature and the amplitude of the provoked response. The heat plate assay shows that larvae exhibit a stereotyped sequence of nociceptive behaviors that depend on the temperature of the surrounding water and the activity of the nociceptive sensory neurons.
The methods described here can help answer key questions in the thermal nociception field, such as, do animals respond differently to noxious thermal stimuli that are presented either locally or globally?
Generally, individuals new to the heat probe assay will struggle because mastering the assay requires practice to minimize variation in the pressure, the location, and the contact angle of the applied probe.
Visual demonstration of the heat plate assay is critical as behaviors elicited with increasing temperature need to be seen to be best appreciated.